Drinking more than 21 alcoholic drinks per week for men or 14 for women is considered excessive and increases the risk of permanent liver damage. The alcohol in beer, wine, and liquor is called ethanol, a small amphipathic molecule that can easily pass through cell membranes, including the blood-brain barrier, leading to its well-known effects on the nervous system. But in this video, I'll focus on the toxic effects of ethanol on the liver. Alcohol is mainly absorbed in the small intestine, but a small amount is also absorbed in the stomach. Either way, ingested alcohol does not directly enter the circulation, but is first transported to the liver through the portal vein. Upon reaching the liver, ethanol diffuses easily into hepatocytes, where the enzyme alcohol-dehydrogenase converts it to acetaldehyde in the cytoplasm. At the same time, a molecule of NAD+ is reduced to NADH, a high-energy electron carrier involved in the electron transport chain. In the mitochondria, the enzyme aldehyde dehydrogenase converts acetaldehyde to acetate and reduces another molecule of NAD+ to NADH. While acetate can be safely converted to acetyl-CoA and into the Krebs cycle, acetaldehyde is a toxic intermediate that damages DNA and proteins through the formation of adducts. However, the increase in the ratio of NADH to NAD+ is also harmful because it triggers a shift in lipid metabolism that favors lipogenesis and suppresses beta-oxidation of fatty acids. As a result, triglycerides accumulate within large vacuoles, displacing the nucleus to the periphery in a process called macrovesicular steatosis, which can progress to a more severe necroinflammatory condition called steatohepatitis. Alcoholic steatohepatitis can still be reversed at this point if alcohol intake is permanently stopped, but ethanol also disrupts the integrity of the intestinal barrier, letting bacterial endotoxins slip through into portal circulation. When resident macrophages called Kupffer cells detect lipopolysaccharide, a strongly immunogenic component of the outer membrane of gram-negative bacteria, they release pro-inflammatory cytokines and chemokines that recruit immune cells to the liver. This inflammatory condition is called alcoholic hepatitis. It's characterized by histological changes such as ballooning degeneration, aggregation of misfolded proteins called Mallory-Dank-Hyaline inclusions, and centrilobular necrosis caused by the death of hepatocytes around the central vein. While still reversible, pro-inflammatory signaling, oxidative stress, and the presence of acetaldehyde eventually activate hepatic stellate cells, which transform into myofibroblast-like cells and begin remodeling the extracellular matrix into fibrous scar tissue. A first collagen is deposited near the central vein, forming a delicate chicken wire pattern. But eventually fibrous septa extend all the way to the portal tracts or connect adjacent lobules, causing bridging fibrosis and isolating small patches of hepatocytes in the process of nodular regeneration. Blood flow through the liver is impeded, causing portal hypertension and the development of ascites and esophageal varices, as portal blood seeks out a return path to systemic circulation. This condition, now called cirrhosis, is irreversible and may require liver transplantation. The flow of bile is also obstructed, causing bilirubin to accumulate in the blood, resulting in jaundice. Over time, the ability of the liver to perform its many essential functions is compromised, resulting in hepatic insufficiency, delayed coagulation, and hypoalbuminemia. Failure of the liver to detoxify ammonia into urea can result in hepatic encephalopathy, in which swelling of the brain causes confusion, disorientation, personality changes, and eventual loss of consciousness. This combination of chronic inflammation and repeated cycles of cell division and repair in the presence of cytotoxic chemicals is dangerous and significantly increases the risk of cancer and liver failure. While the liver can metabolize small amounts of alcohol over time, the perceived benefits of alcohol are mainly social. Ethanol and its metabolic intermediate acetaldehyde are classified as carcinogenic toxins, and no amount is considered safe.